Hello AP Bio students and welcome back to Lab Diaries from AP Bio. In this series, we will cover different lab expectations and review key labs.
This is our final episode, episode 6, DNA Fragmentation Lab. Lab Overview. Problem. How can we use DNA fragmentation to compare different samples from a crime scene? Null hypothesis. No observable difference in banding patterns of the agarose gel because the variations in fragment sizes will be due to random chance. Hypothesis. If a restriction enzyme sample is added, then the DNA samples will show activity in the progression towards the opposite pole and there will be variations in banding patterns. Materials. Things needed for the experiment. 5 tubes labeled as CS, RE, S1, LD, and S2. Micropipettes and tips, a timer, goggles, agarose gel, electrophoresis buffer, a gel tray, sample wells, a power supply, a UV transilluminator, UV protected goggles, and data sheets. The dependent variable in this lab are the distinct bands under the ultraviolet light. The independent variable is the concentration of enzyme used in agarose gel.
The procedure. Set up the restriction enzyme digest. Add 4 microliter of restriction enzyme RE to each of the 3 samples, CS, S1, and S2, by adding it to the DNA sample at the bottom of the tube. Remember to change your pipette tip for each of the sample. Incubate the 3 samples at room temperature for 5 minutes for the enzymatic reaction to occur. Add 4 microliter of loading dye into each of the 3 samples by adding it to the bottom of the tube. Remember to change the pipette for each sample. Centrifuge samples for 1 second. Make sure to arrange them to make it microcentrifuge balanced. After that, load 20 microliter of each of the 3 samples into the wells in the gels. After all samples have been loaded, the teacher will add the electrophoresis buffer to the gel box and cover with lid. Connect it, plug it, turn it on, and adjust to the correct setting to make sure the gel runs properly. The gel will run for 30 minutes at 100 volts. After the gel is done running, the teacher will turn off the power supply and remove cover. Slide the gel off the UV transilluminator and then close the lid of the UV transilluminator. Make sure to wear UV goggles. Analysis. How did it go and our personal results? Having steady hands while using micropipette. This lab requires you to use a micropipette. We recommend that all group members practice on practice gel using micropipette, and after everyone has had their turn, get the person with the most steady hands in your lab group to use the micropipette when doing the actual lab. To get good results that can be analyzed, it's important to practice putting solution in the agarose gel and learning how to use the micropipette. Comparing results with other groups will help you understand the results better. Some lab groups might get clearer results than others, which can be beneficial when observing and analyzing data. Make sure to use UV sunglasses so you can avoid damaging your eyes. I hope our advice can improve your lab experience. Good luck on your AP Biology journey. Thank you for your time.